So the question for my project is, do beta trans-bergamatine floral emissions dictate bumblebee floral preference regardless of flower type? So the main takeaway for my project is that bee pollinator preferences for flowers that transmit beta trans-bergamatine is dependent on flower type. This goes against the hypothesis that if bumblebees were exposed to different flowers, then the bumblebees will respond to flowers that transmit beta transbergamatine irrespective of flower type. So this is a figure generated from my data, and the main findings is that greater amount of bees chose sterile over fertile flowers that emitted high levels of the volatile, and the greater amount of bees chose fertile over sterile flowers that emitted low levels of the volatile. And Grandma Bees chose fertile over sterile flowers that had absent levels of the volatile. And since I used categorical data for this project, I chose to do a category analysis of the data and got a p-value of 0.002, which is less than the alpha level of 0.01, which tells me that bee pollinator preference for flowers that transmit beta transbergamatine is dependent on flower type, which is contrary to the hypothesis. And why matters? Exploring the effects of floral components on pollinator preferences is important for a better understanding of plant-pollinator interactions, which affect the overall health of the ecosystem. In other words, if flowers are not able to attract the right pollinators, plant reproduction would halt since the transfer of pollen would not occur, resulting in an ecosystem of dying flowers, leading to a lack of certain flowers and therefore plants that otherwise support the food supply chain. And we are able to learn about pollinator sensory bias mechanisms in foraging behaviors and the possibility of the presence of certain floral volatiles that dictate innate preference regardless of plant type. As a result, it's able to be seen how plants can take advantage of this bias as a cheating mechanism in order to attract certain pollinators. So this is paper 1. The left shows the Excel of the data set as well as the figure from the article itself. And the data extrapolated from this article comes from this figure, which shows choice of bees when presented with high or low levels of beta transbergamatine from flowers, and the choice of bees when presented with high or low levels of the volatile from volatile samples, where flowers represents fertile flowers while volatile samples represent sterile flowers. And I manually calculated how many bees chose high and low for each sample type to get the total and converted this to a percentage of high low of bee choice. And this is paper 2. The Excel sheet as well as the figure itself is on the left, and the data extrapolated from this data come from this part of the figure, which shows the choice of bees when presented with fertile or sterile antlers without beta transbergamatine transmission. And actually, the part of the data set where it says the volatile is present in the second column, and manually calculated the number of bees that chose fertile over sterile for the total and percentage of each. And I should add that the Excel sheets that I showed were only an example of part of the dataset and does not show all of the dataset. And thank you for listening to my presentation.